Hi, you guys. It's Miss Arneson. I'm in the classroom, which is why I have this mask on. And I'm going to help you with Science 122, which is the designer babies, with Lesson 2, what is genetic engineering? So in order to do this, of course, you need to go to your classwork stream and scroll down to Lesson 2. When you click on it, it does not have the document that you actually need to work in. So you have to go to View Assignment. And then in the corner over here, mine says missing. So it says I haven't turned it in yet, and that's because I just barely logged in with my account. So I'm going to go ahead and open that document. If yours has already been given a 0 or something, it might say that you've already turned it in. You might have to push make a copy or something. So here's a little comic about genetic engineering, and you just have to answer this question. And so go ahead and do that. I'm not going to read all the directions for that part. Okay, so for gene expression, this is one of the most important parts of all biology. Gene expression means like it basically tells you how your DNA gets turned into you as a person. Because remember, DNA is a molecule found in the nucleus of your cells for most of the time, but you are made of proteins. So how do we get from point A to point B or from information into an actual functioning body? And that process is called gene expression. So there's an Edpuzzle video with protein synthesis for you to watch, and the questions are already on there. You can just answer them as you go. Okay, so for junk DNA, I made a fact sheet for you guys. And so this is something that I put together with information from a couple of different sources so that you could kind of understand a little better about what junk DNA is. Oh dang, Las Montañas. It should be viewable for everyone. So I might have to go change that because you don't need to edit it, you just need to be able to view it. So I'm going to go change that setting in case this is giving you troubles. I'm going to pause for a second and go change it. Okay, guys, I fixed the link, but sometimes the link doesn't update on a document that you've already started editing. So there's another way to get to it just in case you need to. And that is if you go to the lesson, all of the attachments for that lesson, it's this one that says non-coding DNA fact sheet. So that's where I'm going to click it. And it has some pictures on here. This is actually a picture of some chromosomes in alignment, and it has different genes highlighted. And then down here, there are some different, the A's, G's, T's, and C's that line up in DNA. That way you can kind of see what they look like up close in their molecular form. One of the things I want to point out is that when you're looking at an article, or this is something that I really condensed for you. So there's a lot of information on junk DNA, and I just took it and I put it down into the least amount of information that you possibly need to know to pass the class and probably to get through all of your exams for high school. So this is just 4 paragraphs, it looks like, of information. But one of the things you want to look at is when there's some highlighted text like this in a text box. This one says, so junk DNA is not exactly trash. Some of it has some use, and we probably do not fully understand all of its roles. That's like a huge point that the author, in this case me, wants you guys to really remember about it. Junk DNA, just the name of it, kind of gives you a good idea of what it is, but it's not exactly trash, so we don't want to completely equate those two. So go ahead and read this, and then there's 2 questions about it that you have to answer back in your document. The first one is, read the fact sheet and take a few notes. Oh, just kidding. So take a few notes that you might need and then take a picture of your notes and put them here. So thank you very much for doing that in advance. You can just paste that picture right here, put your cursor, push enter, and then paste that in. Okay. This part is something that I think a lot of students got bored looking at, but one of the things I really want you guys to understand about science is that scientists don't just come up with these ideas randomly. We do like a lot of research, we put in a lot of effort, we have a lot of different people involved before we're going to say something is true or not. And that doesn't happen always on the internet, so it's easy sometimes to think like, oh, well, scientists just think blah, 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 when no, there's actually a lot of detail and expertise that goes into it. And this is one of those examples. So it is a Edpuzzle and TED Talk. The reason I put it in Edpuzzle is just so that I can tell if you watched the whole thing. I know that's annoying for you guys, huh? but it's a great tool for teachers. So this person is really important in the history of CRISPR, which you're going to learn what that is. And this is a W5S2 activity. So when you do a W5S2, you are looking at the who, what, when, where, and why of the document. And then there's two parts for S, the synthesis, and the significance, or the summary is what it's called right here. So you have to watch the video. First, you put the title of the resource. So what is the name of the YouTube video? Who made it and when was it made? So those things you don't really have to have in complete sentences, but the rest of this you do. So who is the subject of this source? Or sometimes, what is the subject of this source? Who is it about? And so if we go to the video, it at the top says Jennifer Doudna, and it's called How CRISPR Lets Us Edit Our DNA. So is it about her? It's about her research. So honestly, I would not put it's about her. She is the speaker, but what she is talking about is the important thing here. What is it about? What happens? So then you're going to look and you're going to see, well, what is she telling you about? When does this happen? Now, a lot of students are tempted to put the date the video was made, but you do not want to do that when it comes to these, because you're talking about when is the video about more than when was it made. So when is CRISPR happening? When is CRISPR allowing us to edit DNA? Where does this take place? Well, I'm sure you could look up where this TED Talk was, but that's not where CRISPR is. That's not where our editing of DNA is. And so that's what you want to be talking about in that question. Why? Why did this happen? Why did the author or creator make it? So why was this video made? What was the purpose of it? And then a summary is just 2 sentences or less. Like if you can do it in one, I think you should try, because summaries are really important, and students a lot of times want to focus on the details, but the summary focuses on the big picture. All right, the synthesis or the significance is how does it relate to your own life or your community? And you do have to write at least 2 sentences here because I want you to be as specific as you can. How does the ability to change DNA with CRISPR affect your life or your family's life? That's what you need to answer at that bottom part. And you can think of 2 things, and that way you have 2 sentences, one for each thing, or you could write a little more detail about one thing and that would be enough. Of course, if you write extra bonus lines. Okay, a lot of students really still are not doing the essential question connection right. So I'm going to go over that real quick. Essential question connection means that you need to pick one of these 3 questions. These are our big essential questions for the unit. For everything we learn in this class, you should be getting an answer to one of these questions. So is this one about how genetic information is coded for in DNA? About how DNA changes over time? That would be like kind of an evolutionary thing. Or you could talk about it in the case of the CRISPR. And how can scientists predict gene and trait frequency? So we didn't really talk anything about genes or traits. So you could probably use one of these top 2, and all you have to do is explain how you have a part answer to one of these questions. So you do not have to copy and paste the question. You just need to highlight the one that you think is correct. Okay, I hope you guys have a beautiful day, and let me know if you need any more help.